In this lecture, we are going to discuss the bioinformatic tools for searching in sequence databases. These tools allows a researcher to compare a query sequence with other sequences in the database and identify those that are similar to the query. As we discussed in previous lessons, sequence similarity is indicative of evolutionary relationship between genes and proteins, helping to the functional and structural assignation between related sequences. So, if we know that two sequences are similar, we can make a hypothesis about their common origin, biological function, and general structural features. So far, we have been seeing the different methods for the comparison of two sequences, either nucleotides or amino acids, using global or local alignment techniques. These methods require previous knowledge of the sequences to be compared. But what happens if one has an unknown sequence and wants to investigate it? We already know some basic terminology used in the sequence comparison field, like identity, the similarity, the homology, and the two principal methods for the pairwise sequence comparison, as the global and local alignments. Now, we include some new terms for the case of the search of sequences. The query, a sequence of nucleotides or amino acids to search for. The subject, the database against which you search. And a hit, a sequence that matches the query. Sequence similarity search is a very important tool in the genomics and proteomic fields, helping to the discovery of regions in genes or proteins that are essential for the function. This is a schematic representation of the process of searching for similar sequences. As input, we have the sequence to be investigated. We need an algorithm to perform the comparison of the input sequences with the sequences stored in the database, and an output where we obtain the results of the sequences that match or are similar to the query sequence. For doing this type of searches, there is a specialized program called BLAST that uses an heuristic local alignment approach to find sequences similar to a query sequence. The basic local alignment search tool, or BLAST, is specialized in finds region of local similarity between sequences. The program compares nucleotide or protein sequences to other sequences stored in a database and delivers the sequences in the database that are similar to the sequence consult. How BLAST works. This is the basic scheme for the web server model of a BLAST search. The user submits a query sequence to the BLAST web page, and the BLAST server engine finds those sequences similar to the query in the database, and then return the results that you can see in your screen. As you can see from the scheme, the search engine and formatter are decoupled, so it is possible that one can view a variety of different output formats and only perform one search. Why to use black? We already know various methods for sequence comparison as global or local alignments. Unfortunately, these methods are too slow to be used for the comparison of a sequence with many sequences, usually millions, stored in a database. That's why we need a new algorithm to speed the process. BLAST uses an heuristic Smith-Waterman algorithm which seeks local alignment and is therefore able to detect relationships among sequences which share regions of similarity. The use of an heuristic method in BLAST allows to find similar sequences not by comparing either sequence in its entire length, but rather by locating short matches of coincidence between the two sequences. Let's take a look to the principal steps used in the heuristic of a BLAST search. There are three main steps: the seeding, which is defining initial words in the query sequence with high score in the sequence database, then the comparison of high score word lists to the database and identify exact matches, and third, to make alignments using a Smith-Waterman approach. Let's go to see the steps. The first step, the seeding step. The first thing is to divide the query sequence in a small part. In this example, 3-letter words. Three-letter words are usually used for protein sequence, whereas for nucleotides you can use large words, 7 up to 15 words, depending on the sensitive that one desires. Once we have the query sequences divided in small words, we compare all the words we generate from our query with a list of words of similar size, in this case 3, that are included in the database. And we select the words that are more similar to the query using a score threshold based on a substitution matrix. Words in the database that compile and score above this threshold of similarity are discarded, and words that compile and score above the threshold of similarity are selected for the next step. In the second step, we find the exact location in the database sequences of the words that best match our query sequence. Sequences in the database that do not contain any such word are discarded, and sequences that contain the high score words are considered hits and are selected for the next step. The final step involves the alignment of the query sequence with the selected sequences, the hits, in the database using a local alignment approach. The alignment starts in the 3-letter matched region and is extended in both directions, calculating the score every time a new letter is aligned. The alignment stops when, with the addition of a new letter in the alignment, decrease the score of the alignment below a predetermined cutoff. These alignment segments are considered high-scoring segment pairs. Once this process is complete for each sequence in the database, a report is generated that includes the resulting alignment. Here we have a summary of the BLAST heuristics. This process is very fast, but you can miss solutions in the seeding steps and in the selection of sequences to be compared. Moreover, the heuristic search only found close matches which should be quantified to estimate the significance level. That's why we need good statistics to evaluate the significance of the encountered solution. We already know that the raw score formula for evaluate the alignment of two sequences. This type of score depends on the parameters for matches and mismatches obtained from the substitution matrix and the gap opening and extension penalties employed in the search. If we can compare results of different BLAST searches, we need to normalize this scoring system. That's why, in similarity searches, a bit score is more useful. By normalizing a raw score using this formula, one attains the bit score, which has a standard set of units. As a result of the normalizing process, bit scores are independent of the scoring system, allowing those calculated with particular rewards and penalties to be compared directly to those calculated with different rewards and penalties. Bit scores are normalized, which means that the bit scores from different alignments can be compared, even if different scoring matrices have been used. The second, and perhaps the most important statistics in a similarity search is the E-value, or expectation value. A BLAST search returns hits, sequences that produce significant alignments to the query sequence. The significance of a hit is measured by its E-value or expectation value. Expectation value are used to assess whether a given alignment constitutes evidence for homology and helps to know how strong an alignment can be expected for chance alone. E-values are calculated from the following three factors: the bit score, or the normal score, the length of the query, and the length of the databases. As you can see from the formula, since a large score is less likely to be obtained by chance than a smaller score, larger bit scores correspond to smaller E-values. In the case of the length of the query and the database, A particular score is more easily obtained by chance with a longer query or database size than with a shorter query or database size. So, longer queries and larger databases results in larger e-values. Thus, the e-value is actually a statistical parameter which accounts for the significance of an alignment. The lesser the e-value, less is the probability that the alignment is a result of mere random chance. In general, good e-values tend to be very close to zero. In other words, an e-value of 0 for a determinate alignment means that there is no chance to obtain this particular alignment by chance. Now let's go to the practical aspects of a BLAST search. These are the basic steps for a similarity search in a database using BLAST. Step 1. Select the BLAST program. First, the user has to specify the type of search to be conducted. There are several options, depending on the query sequence, if it's a protein or a nucleic acid, and the biological problem we are interested for. These options we will discuss later. Second, the user has to provide the sequence for the similarity search. For this, there are a number of formats that are accepted by the program. Third is the selection of the database for conducting the search. In this case, we have a number of databases for select according to our biological problem. Those databases for nucleotide sequences includes gene bank sequences of nucleotides, genomic reference sequences, chromosome sequences, expressed sequence tag sequences, high throughput genomic sequences, patented sequences, contigs of genome shotgun sequences, and others. In the case of protein sequences, we have the sequences of proteins that are included in the gene bank, reference protein sequences, the non-redundant UniProt knowledge base and SweetProt databases, sequences of the protein data bank, and sequences came from genome shotgun projects, patents, and transcriptome. The first step is the selection of the algorithm and parameters for the search. There are several algorithms depending on the nature of the query sequence and the biological problem we are interested to solve. Parameters like e-value, the word size, the query range, the scoring values, that means mismatch and cut penalties, and filters could also be customized. The final step is the submission of the BLAST search according to the select database and algorithm parameters. Now let's go to analyze the result of a BLAST search. The BLAST result on the NCBI side is divided in three main parts: a graphical display, a collection of hits, and the alignment corresponding to the identified hits with the query sequence. The graphical display represents in a visual way the obtained results. The top bar stands for the complete length of the query sequence. Colored bars below the query represent the sequence founded. The position and length of the bar indicates the region of the query sequence that a high score segment pairs cover. And the color represents the score obtained by this alignment. This is the scale, color scale. The second output is the hit list. The hit list is a table of all identified sequences in the database that match the query sequence. They are usually organized by expectation value. In this case, the three first hits are exact matches of the query sequence, which means that it's the same protein. Also, there are listed the score values of the corresponding alignments of all hits with the query sequence and the regions that covers from the original sequence. These low values represent these small fragments that were obtained from the comparison of the sequence. Finally, we have the alignments of our query with each of the sequences that match significantly our sequence. Each alignment has a bit score associated, which is a measure of similarity between the hit and the query. The e-value obtained by this alignment, and also there are other metrics like the values of identities and similarities, as well as the number of gaps. So, for we consider a biological significant hit, we need to obtain lower e-values, high scores, and high values of identities and similarities in the alignment. We already mentioned that according to the biological problem, we can select several alternatives for a BLAST search. Let's go to discuss some of them. There are different types of BLAST searches. On one hand, we have the basic BLASTs, with different options for the treatment of the sequence and the database to compare. In this category, we have the BLAST-N, the BLAST-P, the BLAST-X, the T-BLAST-N, and the T-BLAST-X. In addition, we have specialized BLAST for more specific problems, like defined primers specific to a PCR template, or find a conserved domain, or search for sequences that have gene expression profile, and others. In this view of the NCBI BLAST webpage, you can choose the type of search you are interested for. The basic BLASTs and the specialized ones. And we have also options of different algorithms we are going to discuss later. These are the typical and basic use of BLAST for searching for DNA and protein sequences. BLAST-N for nucleotides and BLAST-P for proteins. These are the most common and more employed searches for proteins and nucleic acids. There are other BLASTs, type of BLAST, like the BLAST-X, that search protein databases using a translate nucleotide query. Because BLAST-X translate the query sequence in all 6 reading frames and provides combined significant statistic for hits to different frames, it is particularly useful when the reading frame of the query sequence is unknown or it contains errors that may lead to frame shifts of other coding errors. So this type of BLAST is very useful if we want to discover proteins encoded in a query DNA sequence. The T-BLAST-N search translated nucleotide database using a protein query. ESTs, or express sequence tags, are short, single-read cDNA sequences. They comprise the largest pool of sequence data for many organisms and contains portion of transcript for many uncharacterized genes. Since the ESTs have not annotated coding sequences, there are no corresponding protein translation in the BLAST protein database. Hence, a T-BLAST-N search is the only way to search for these potential coding regions at the protein level. Like all translating searches, the T-BLAST-N search is especially suited to working with error-prone data like ESTs and draft genome sequences, because it combines blood statistic for hits to multiple reading frames and thus is robust to frame shifts introduced by sequencing errors. The T-BLAST-X effectively performs a more sensitive BLAST-P search without doing the manual translation. T-BLAST-X gets around the potential frame shifts and ambiguities that may prevent certain open reading frames from being detected. This is very useful in identifying potential proteins encoded by single-pass read ESTs. In addition, it can be a good tool for identifying novel genes. TBLASTX is computationally intensive, so thus for searches of large genomic queries is not recommended. In general, for users with regular needs for this type of searches, the best way is to install a standalone BLAST and perform the search locally. Here is a summary of the basic type of BLAST. We could search a protein database using a protein query. We could search a nucleotide database using a nucleotide query. We could search in a protein database using a translate nucleotide query. Also search a translate nucleotide database using a protein query, or search translated nucleotide databases using a translate nucleotide query. In addition to the basic and specialized type of BLAST searches, one can employ different modification of the BLAST algorithm for special purposes. This is the case of MegaBLAST and discontinuous MegaBLAST for nucleotide sequences for search high similarity or more dissimilar homologous sequences. On the other hand, in the case of proteins, we have several algorithms to improve the sensitivity of the search. That is the case of PSI-BLAST, PHI-BLAST, and DELTA-BLAST. These modifications respond to the modular nature of proteins in domains, which are often identified as recurring units, which may exist in various contexts. In molecular evolution, such domains may have been used as building blocks and may have been recombined in different arrangements to modulate protein function. That is why modification of the original BLAST algorithm, we can detect the presence of these domains in distantly related proteins. Let's go to see through an example. Let's take an example of the application of one of these methods for the detection of remote homologs of a given protein family, which could be characterized by the presence of one functional domain. In this case, we are going to see the example through the zinc finger proteins. Zinc finger proteins are proteins that display a particular structural fold that is essential for the coordination of one metal element. There are many proteins from different organisms that belongs to this family of zinc coordinated proteins, and many of them are not necessarily easily related by a simple inspection of the scores between their alignments. In many cases like this, functional and evolutionarily important protein similarities are recognizable only through comparison of the three-dimensional structure. But when such structures are not available, patterns of sequence conservation identified from the alignment of related sequences can aid to the recognition of distant similarities. In this case, we observe that when we obtain a collection of alignments of related proteins, there are some patterns that are conserved. Even if the rest of the elements of the sequence are not. In this case, the presence of two cysteines and histidines amino acids which are essential for the coordination of the zinc ion are the conserved elements. Thus, it's desirable to have methods that allows to extract the essential patterns of a group of sequences and use it for the detection of other sequences that in many cases are so distant in evolutionary time that are difficult to detect by conventional methods. There is a large literature of the definition and construction of these patterns, which have been named as motif profiles, position-specific scoring matrices, and hidden Markov models. One of such kind of algorithms to detect remote homologs is the SciBLAST, or position-specific iterated BLAST. This method utilizes a constructed profile for a group of related sequences for the search of other distantly related, attending to the principle that in spite of the low overall conservation between distantly related sequences, there will be some regions or patterns of high conservation generally implied on the functionality. How it works. There are four main steps in a SciBLAST search. These are: run a standard BLAST search using a standard substitution matrix. With the search results above a specific threshold, construct a position-specific scoring matrix, and next, search in the database using this position-specific scoring matrix instead of the initial and general score matrix. This process could be repeated many times until obtain some results or no new sequence are found. Let's go to see how SciBLAST works. The first step is conduct a standard BLAST search for the query sequence using a standard scoring matrix. These are our query sequence, and suppose that these represent the pattern of conservation, and make a normal BLAST search. In this case, we are talking about a protein, so we use BLAST-P and Blossom 62 as scoring matrix. We obtain a number of solutions. From these solutions, we collect those that score about a specific threshold that we select. These alignments with high scores that conserve the property are selected. Other alignments with bad scores are not selected. These alignments of the sequences in the database that obtain a score above a specific threshold are used to compute a position-specific scoring matrix. These matrices are analogous to the classical substitution matrix, with the difference that the ways of changes or substitutions are specified according to the position in the sequence. This is a position-specific scoring matrix for the example that we are discussing, of zinc finger proteins. Here you can see that in the first column is specified the location of the elements in the sequence. This is the number of residues in the sequence. Whereas the row represent the 20 amino acids. As we observe, the cysteines and histidine residues that are involved in the binding of the metal, which is important for the functionality of this type of proteins, have higher weights than the rest of the elements in the sequence in these positions. Higher values for the histidine to be conserved and for the cysteine to be conserved. A difference of other cysteines or histidines that you can find in the rest of the protein sequence, but they are not in the correct position for coordinate the metal ion. So this conservation is represented in a matrix like this. The process to perform a SciBLAST search is iterative. First, a list of closely related proteins is created using a standard BLAST search. The first step is doing a normal BLAST search and collect the first high score alignments. These proteins then are combined into a general profile sequence which summarize the significant feature present in these sequences, and then a second query against the protein database is conducted, but instead of using a normal substitution matrix, we use the position-specific scoring matrix. After this second iteration, we obtain new matches in the database, and with these new matches, if they have a score greater than the threshold that we established, we update our position-specific scoring matrix and then run another BLAST search using the update matrix and obtain new result. And continue this process until we obtain a good result or no new sequence is found. This is the way that the SciBLAST works. These are the basic concepts of a similarity search in sequence database using BLAST or the different BLAST tools. The practical exercise of this lesson will correspond to the search for the searching of homologous sequence to a query using the NCBI Plus web server.